The title of this presentation is Innate and Adaptive Immune Cells in ME/CFS, and looking at the current research that I lead along with my colleague Dr. Don Staines, where we established the National Centre for Neuroimmunology Emerging Diseases. So this is our centre here. And you'll notice it's in purple and blue because of the 12th of May in the International ME/CFS Day. So I'd like to, at this point in time, acknowledge my students and my postdoctoral research fellows for their contribution to the research that I'm about to present. And the research that I'm presenting today is outlined in 4 studies, outlined here in Study 1, and that is I'm going to present specific immunological data, that is natural killer cell lysis in severe and moderate CFS-ME patients, as well as particular receptors that govern the lysis activity. And then following it up with Study 2, where we obviously needed to repeat this study in a larger cohort, and for this reason, Study 2 is going to focus on not only natural killer cell lysis, the receptors that are involved with that lysis activity, and if you're not sure about lysis, I'll explain that in the subsequent slides. And following, I'm also going to present data in Study 2 in relation to specific cells, and that is the title of my talk, Innate and Adaptive. So the cells that are going to be covered are going to be part of the innate immune system as well as the adaptive. Study 3 is looking at particularly natural killer cells, and what we're looking at in this instance and the data I'm going to present is focusing on not only lysis but also the mechanism of how lysis occurs. And for that reason, that data will be presented in different stimulative cells, and I'll outline what those cells are and the effect of the stimulation, that it'd be representative of an antigen stimulation. Finally, Study 4 is looking at the submolecular components that control not only white blood cells and the proteins that are produced, but all genes that are transcribed within our body. So those submolecular controlling mechanisms, for example, are called microRNA. And this study looked at microRNA, and we focused on plasma. Okay, and we used high throughput sequencing, and that's been mentioned today. We've completed this study, and it's showing there's 3 favourable microRNA that potentially now we need to validate for maybe biomarker detection. So I'll talk about that at the latter part of this study. And then Study 5, if Ian permits, hopefully I'll have my 40 minutes by then, and that is looking at we're focusing on the prevalence of CFS-ME because there's only been one study, and that's been done by the Dubbo study. The definition, if you're aware of that Dubbo study, is quite controversial. And for this reason, we've focused on the CDC and the ICC criteria, and we're looking at the actual prevalence of CFS-ME through our generating our own prevalent data using such questionnaires, and this is through an actual online database I've created. But more recently, and I think this is something that will resonate with the audience here today, is I'm going to show an iPhone app that I've developed in conjunction with my colleague that will give a weekly account for patients to record their symptomology as well as be able to PDF that document and keep it as a weekly record of their signs and symptoms, and subsequently they're able to then email that to their physician, and it gives the physician an accurate account of their history. So, the title of this presentation is Innate and Adaptive Immunity, and the cells that I'm going to cover today are highlighted in these particular red boxes. That is, we're going to focus on this innate component here. Dendritic cells, and they act as antigen-presenting cells. In the presentation today, you'll see there's a number of different dendritic cells. They have different phenotypes, and they've all got different functions. So I'm going to highlight those functions and what we found. In addition, there's been a large amount of literature that has investigated natural killer cells. And our group has shown significant changes, not only in lysis, but in cell receptors that are expressed on these particular cells, which govern their function. So we're going to look at these cells, and we're going to go through the receptors in the severe group as well as moderate CFS patients and show potentially differences in the function of these NK cells in the severity of the illness ME/CFS. Now, what you're going to see also is I'm looking at particular B cell phenotypes, and the rationale for that is memory B cells, as you've heard today, they produce antibodies, and obviously if you've got an increase in memory B cells, that might suggest there's an autoantibody. So I'm going to show the particular phenotypes we've investigated in these studies and those results and how different they are with particular phenotypes for both the severe and the moderate CFS patients. Now you'll notice that these two cells here are an overlap. They overlap between the innate and the adaptive, and these two cells are called NKT cells and gamma delta T cells. They function with lysis activity similar to natural killer cells, as well as they have antigen presenting capability. And the data I'm going to show is in relation to the varying differences of these particular cells and what does that mean in clinical signs and symptoms for severe patients as well as moderate. So just briefly, you all would be aware of this cell, the natural killer cell. The study, the two studies, study 1, 2, and 3, I'm going to present, is not only looking at natural killer cells and how they kill or lyse the particular target cell represented down here, but I'm going to present data in relation to receptors that are expressed on the cell surface of the natural killer cell. And these cell surface receptors are a number of groups. They're called killer immunoglobulins. They either activate or they inhibit natural killer cells to then obviously induce cell death. Now, how they activate or inhibit, that's yet to be elucidated, but we're characterising those particular receptors. And in addition, how they kill is due to this cytoplasmic granule outlined here. Now, around this cytoplasmic granule are these particular proteins. They're called CD107a and CD107b. They encapsulate this cytotoxic granule, and then they move it to the cell membrane where it makes contact with this target cell, and they basically then induce lysis. So I'm focusing on those because they're seen as the critical component in terms of how a natural killer cell is able to move these proteins, which are very hostile, across into the target cell and lyse and kill that cell. Okay? So in terms of these natural killer cells, not all of them, if we go back to here, not all of them do this particular function. There are two particular phenotypes of natural killer cells outlined here. This cell here, the natural killer cell, is called a CD56 dim cell. The key information you need to take from this presentation is the CD56 dim cell, its main function is lysis, like I showed in the previous slide. However, this phenotype of this natural killer cell here, represented on the right-hand side, is known as a CD56 bright cell. It has lower killing activity or lower lysis activity, and its primary response outlined here is to produce a number of inflammatory cytokines. So these two main phenotypes are key in terms of what's been investigated in CFS-ME patients and how these have been reduced, and a heightened response in terms of cytokine production has been documented by my group. So, just before we begin about the results, I just want to orientate you to this particular diagram. This diagram outlines, if you look down here at this lower section here, this diagram outlines microRNA. They are non-coding, small, very non-coding RNA, and what they do, if you look here at this diagram section here, and also here, they bind onto the gene that's going to be transcribed, as represented here with the red and along here, and they inhibit that actual gene to be transcribed. So it's important to see whether or not gene expression in CFS-ME patients, whether they be white blood cells or other cells. You've heard about different types of proteins, hormones, etc. today, whether or not microRNA may play a role in regulating how much of a gene is actually produced. So if I take you to this slide, you notice at the start of this presentation I said study 1 was investigating immune and gene changes in severe and moderate patients. Now, the results I'm going to present to you today are primarily on study 1 related to natural killer cytotoxic activity. That is that killing or the lysis activity. And we investigated this late in 2013, and we investigated it in a small, severe, bed-bound, house-bound CFS patient group where we visited with a clinician the patients, and then we had this moderate group and control. What you can clearly see is there's a significant reduction in natural killer cell lysis between the control group and the varying severities of CFS. In addition, if you look at what governs that actual receptor facilitation of reduced lysis, we then looked at the characterization of these killer immunoglobulin receptors, the activating or the inhibitory receptors on these natural killer cells. What we found is this receptor here, KIR3DL1, is significantly increased in the moderate group, and there was no significant difference in the severe, but compared to the control. Now, the key take-home message of this result is that KIR3DL1 is an inhibitory receptor. So you recall we had a significant reduction in NK cell lysis in both the severe and the moderate patients, which means that we are now potentially characterizing a cellular mechanism that may be responsible for such a reduction in the natural killer cell function. So we wanted to repeat this, and this is where study 2 then follows suit. So study 2 I'm going to present to you is in terms of looking at the reproducibility with a different severe group, as well as looking at the natural killer cell receptors, and then following it up with dendritic cells, etc. So the take-home message from this actual slide is there's a significant difference between the control and also the moderate and severe group for natural killer cell lysis, and these are different target ratios for this cell that we're actually making the natural killer cell target, but it's still the same uniform significant reduction. We followed it up, and I've put this over here just to remind you of the two different phenotypes. So this here is the DIM phenotype, and we looked at the different receptors on the natural killer cell. And what you can see is this KIR2DL1 was significantly reduced. Okay, so what does that mean? This DIM cell is responsible for large amounts of cytotoxic granules. So this means this stimulatory receptor, the one that would be stimulating to activate the natural killer cell to then induce lysis, is significantly reduced. So we then looked at the DIM cell, the natural killer cell. Now the DIM cell, if you recall, is responsible for the high lysis activity in basically eliminating infected cells. And we looked at this additional marker here. It's called CD94, and it's located on this natural killer cell. What we found was there was a significant increase in this CD94 protein expressed on this natural killer cell DIM cell. So this means the increase, a significant increase in this CD94 receptor, it is actually responsible for reduced lysis. So the higher the expression on the cell surface, such as in the severe and the moderate, is a representation of a diminished NK cell lysis. We followed up with looking at dendritic cells. No one's looked at dendritic cells. Dendritic cells are very important. They actually are part of the first line of defense of processing and also then presenting the antigen to the adaptive immune response. And what we did is we looked at the different phenotypes of these dendritic cells. What you can see here is there's a significant increase in this plasmacytoid dendritic cell, as well as this particular dendritic cell phenotype. The take-home message from each of these significant changes is, firstly, this plasma dendritic cell, it's responsible for initiating natural killer cell lysis. So in these two groups of CFS-ME patients, potentially it's in a hyperactivity. It's trying to promote the lysis activity through cell-to-cell contact with the NK cell, but it seems not to be actually achieving that overall goal. That is, it's not achieving enhanced NK cell lysis. Now over here on this diagram here, what you see is there's a heightened, a significant increase in this particular phenotype. The function of this particular phenotype is it produces large amounts of cytokine, pro-inflammatory cytokines, and these phenotypes activate B cells and natural killer cells. So this cytokine elevation that is significant may be a manifestation of how severely affected those clinical signs show in the severe CFS-ME patients. Finally, if we look here, and we've heard today about B cell memory cells, etc. This presentation also investigated B cell phenotypes, and this is the third time we've reported such a phenotypic change, and that is we looked at memory B cells, we looked at naive cells and transitional cells coming from the bone marrow. Naive B cells are those that haven't previously been exposed to antigen, and memory cells, they reside in the lymph node, the germinal centres, and hopefully that's a programming of protection. You've got antibodies there from prior exposure. The take-home message from this slide is, first and foremost, there's a significant increase in memory B cells. So there's a priming of the memory B cells for antigen that's been previously seen. Whether or not that's an autoimmune indicator, that's yet to be elucidated. But nonetheless, this plus other research groups confirm such a result. And again, it's in the severe group compared to the moderate group. So it might mean a heightened autoimmune response. If we look here at the gamma delta T cell phenotypes, what we can see here is there's a significant difference here in this particular gamma delta T cell phenotype, and likewise over here on the right-hand side. The significance of this reduction in both these phenotypes is important in relation to the function of these particular T cells, and that is this gamma delta T cell phenotype as well as this particular one here, they're responsible for lysis of antigen. So of course you've got a significant reduction which now paints a greater picture of what's actually happening within patients that have CFS-ME, and that is they've got further compromisation in these other cells that perform lysis activity. So if we go on and look at these INKT cells, remember they're the overlap between the adaptive and innate. What you see is an increase in the total number of these INKT cells, and then when we break it down over here to this population, you see these three phenotypes here, there's a significant increase. Now what's the function of those particular cells? They're responsible for producing significant amount of inflammatory cytokines. So I draw your attention to this particular result here. This phenotype's the predominant one versus these two, and in the severe group we have a significant increase in this actual phenotype, suggesting heightened inflammatory cytokine production. So finishing with some other results here, what we did was we isolated these natural killer cells and we challenged them with particular antigen stimulation. And what you can see, the take-home message from this slide, first of all, is not to be overwhelmed, but the take-home message is these results here I'm outlining to you suggest that, remember I was talking about the CD107a and the 107b, it packages up the granule to then lyse the target cell. When we challenge the natural killer cell and stimulate it with an antigen, these 107a, 107b proteins are significantly reduced. And that suggests the lysis activity of the natural killer cell. We can now potentially propose that the actual envelope that contains these proteins that lyse the target cell are actually being produced in a lower amount. And finally, if you look here, when a natural killer cell kills a cell, it's like a bloodhound. It's got to traffic to that actual site. The bloodhound, in this instance, we wanted to measure how effective it is to be able to move to that site to kill that target cell. And we did this by measuring these particular cell markers. They are a marker of how effective this natural killer cell is to sniff out such a target cell. And what you see is there's a significant reduction in these actual markers. And these markers are adhesion. They basically clamp on to the target cell and anchor it there to then ensure it lyses. Subsequently, these results suggest there's a decrease in the migration ability of the natural killer cell to move to that particular infected or target cell, and hence then now you've got even further compromisation in the lysis of the natural killer cell. So study 4 I'm just going to present to you is in relation to high throughput sequencing. So it's a new and novel technology. This is the first to report such results. It's coming accepted into PLOS ONE. What we did is we isolated white blood cells, PBMCs, and then subsequently we then had plasma as well. And we took the plasma because plasma is an indication of what's happening within the cells in the body. So it's a new and novel technology, and we applied this technology to see if we could detect microRNA that would be basically being released from the tissue and see if we could find microRNA that might be a marker of some abnormal tissue function. And we found 22 microRNA, and after placing them through three rigorous statistical phases, we found 3 that are significantly changed in CFS-ME patients. Now, the importance of these microRNA are first and foremost what they control, and that is they're important in preventing inflammation. So there's heightened amounts of them, meaning they're trying to damp down the inflammation response. They are controlling T cell development and Treg function, and our group has previously and continuously reported heightened Treg function. Treg function can be potentially a marker of autoimmunity. So now what we're doing is going back and we're validating these microRNA, because this might be just a possible test that we could use as maybe an indicator for CFS-ME. And we're validating these at the moment through through multiple sclerosis patients, through rheumatoid arthritis patients, as well as some other autoimmune disorders. So to sum up thus far for this presentation, we've found consistent reductions in NK cell lysis in both the severe and the moderate. We seem to see that severity also reflects changes in the immunological function of cells, namely the killer immunoglobulin receptors that are those flags on the natural killer cell. The mechanism of how lysis occurs in the natural killer cell, that is that degranulation through these markers 107a. We find changes in gamma delta T cells, and they are responsible for lysis activity and processing and eliminating antigen. What we've found also is increases in dendritic cells and INKT cells, as well as memory and B cells, and that highlights that there's significant immune dysregulation, whether or not this is due to changes in microRNA that regulate such cells and may be a marker of autoimmunity. That's what we're hunting for at the moment. So this black screen here is intentional, and the intentional delivery of this screen is for me to say something first and foremost, and that is I have taken on board for 5 years since undertaking such research that the patients find it frustrating that they have the inability to accurately record on a continual basis how their symptoms manifest and that cognitive impairment when you go to the doctor and say, oh, I think I was like this last week, but I can't remember what I was like the week before. And my colleague, Dr. Staines, and I have anguished over this for a good probably 3 or 4 years. And driving home just before Christmas last year, I had an insane idea, and that was to invent an iPhone app where patients have the ability to be able to record on a weekly basis their signs and symptoms and be able to recount that in an accurate way. And so I'm using certain questionnaires that we all employ in our research. I've come up with this app with Dr. Staines, and also then it's applied to MS patients because they have the multiple sclerosis impact scale. But the fatigue scale is also used for MS patients, so I can compare. And in addition, we have the rheumatoid arthritis patients, because again, the characteristics of fatigue, inflammation also are a manifestation of that disease. And on that basis, we came up with this app that hopefully, it's going to be released at the end of next month. It's called CliniHelp. So it's a clinical help tool for patients. And what you can see is patients identify with what they are, as in rheumatoid arthritis, MS, CFS, and this is in consultation with neurologists and rheumatologists I've worked with. And this goes through on a weekly basis, not this is the initial details you give, and then subsequently it goes through and gives different signs and symptoms, and you record those on a weekly basis. Now you might say, how does it know to record these on a weekly basis? You get a push notification. So each week you get a reminder saying your time to do this recollection is within this day. And subsequently, what happens is it's an ongoing weekly account of your signs, symptoms, and also it allows then for everyone internationally to be able to use this tool to characterise if there's subgroups on a weekly basis. It allows then whether or not the prevalence of CFSME is varying in different areas, as well as it allows then the patients to have more control and more accuracy in how they're feeling on a weekly basis. And this is the PDF document that you receive, and what you see down here is the section D and E. This is the multiple sclerosis impact scale if you're an MS patient, or a HAQ score, which is done at home for rheumatology patients. And then obviously you can email it to your physician, or you can keep it to yourself, and then when you go to see a physician, you've actually got an accurate recollection on a weekly basis of your illness. If you have a different drug, there's ability here to insert that, and subsequently you can then track whether or not in an accurate manner you've got any improvement whatsoever. Okay? So I think I'll finish, and I finished within the time, so that's a good thing. And I'd like to just acknowledge my colleague who's standing up the back, Dr. Don Staines, the two clinicians I work with very closely, Lisa Beecham, who's a wonderful physician, Chris McClay, the two postdoctoral research fellows that think basically I don't sleep, but nonetheless that's part and parcel of it. And the two consultants, Jennifer Ung, which has the largest rheumatology practice in Queensland, and also Dr. Richard Kwiatek in Adelaide. The research team outlined here, my research students, and this is the website if you would like to go to look at it further. And obviously, research doesn't happen without ongoing funding, and I'm significantly, Dr. Staines and I are significantly supported by the Queensland Government, as well as the Mason Foundation. I'd like to acknowledge Christine Hunter and the Alison Hunter Memorial Foundation, who Richard and Pia work with and is a driving force in Australia. And finally, the Edward P. Evans Foundation. Thank you.
Thanks. Right, time for questions. Questions for Sonya?
I expect that most people in the audience will be non-psychiatrists, but I know that the psychiatrists who hear what you say will say that the immunological changes you describe are downstream of psychological dysfunction. To what extent does that argument still hold water, do you think?
Sorry, I couldn't hear. To what extent?
To what extent do you think the argument that the psychological changes you describe, the immunological changes you describe, are downstream of psychological dysfunction?
Okay, it's a valid question, and I think you're looking at the immunological changes in isolation. However, if you look at it in a holistic manner, what, say, for example, Professor Newton's about to present really doesn't typify or rationale how you can have a psychological change with ANS. If you then look at the other researchers, such as most recently the Japanese group Watanabe, who have shown that there's neuroinflammation, then again, that's the postulation of it to be psychosomatic or psychological. There's no plausible rationale for such a hypothesis.
You alluded to an association with KIR molecules and chronic fatigue.
I think if you look at the KIR molecules, as in whether they're stimulatory or inhibitory, you have to look at first and foremost, not just looking at them in isolation. Okay, it's the proportion of those KIR molecules that are located on each natural killer cell. And in addition to that, you have to look at the HLA, where they're located and the expression of the HLA, because that governs those KIR molecules as well. So HLA typing we're undertaking at the moment amongst other groups. And I think that's something that will probably provide a good rationale for the results that we're finding.
Because, I mean, it's an incredibly heterogeneous and polygenic complex.
Yeah.
So what kind of group sizes are you working with there?
So the group sizes at the moment, we've got a database of 350 patients, all right, that we have as a source, and that's what we've been working with. And they also, may I just add, they meet the CDC as well as the ICC criteria.
Thank you very much. Interesting. I just want to give you a comment because having defects in the innate immunity is a very well-known disease in this group. Lupus do have a depletion and sometimes defects of the C3 and C4.
Yes.
And I just want to comment on that. That's very interesting that you also see some sort of depletion of the NK cells.
I think your comment...
And this is very, very important issue.
Yeah. I think your comment resonates with me, and perhaps next year I might present other results.
Opposite results.